Hello. Today's lecture topic is histograms, but it's actually much more than just making the graph and looking at the graph of histograms, because making a histogram is going to be an incredibly important first step before we can do most of our statistical tests, right? So when we talk about making other kinds of graphs as well as making histograms, they're going to be our first step when we first get all of our data from our experiment. And we're so excited. We've been working on this thing possibly for years. We really want to know what is going on with our experiment. And so we're going to get it, we're going to pop it into a graph and look at what our results are. So when we have different kinds of variables, different graphs will be the right way to do this. So we'll talk about that in another lecture. But for a single quantitative variable, the histogram is going to be one of these exploratory data analysis practices that we're going to do. All right, so this needs to be a tool in your toolkit. And so when I say exploratory data analysis, that is going to consist of graphing the data, making a visual summary of it, like I said, so that we can see what happened in our experiment. We're also going to explore patterns that we see in our data, test hypotheses possibly, but we're really preparing to do the formal data analysis. And we may make a wide variety of these types of graphical summaries, numerical summaries, where we look at things like mean, mode, median, standard deviation, interquartile range. We'll come back to these in another lecture. And when we have quantitative data, that is, we have taken some variable that is a quantitative variable, and we want to look at what our result is. We're going to make this graph called a histogram. Now, histograms are going to look a lot like bar graphs. Bar graphs, however, are for categorical data, and there's going to be spaces between the bars on the x-axis. And for the histogram, it is for quantitative data, and so there's going to be no spaces between the bars. And if you have no idea what that means, we'll come back to it when we're looking in Excel. All right, as we get started talking about making measurements and observing individuals and different variables, I want to make sure that you are using these vocabulary exactly the way we are going to use them for statistics. So when we say individual, it doesn't necessarily mean a single independent person like you or me. It's the thing we're measuring, right? And sometimes our thing isn't a whole thing. We might be measuring circumference of the left paw of wallabies or something. We might be measuring multiple leaves on the same tree. We could be measuring multiple variables on the same individual. So the individual is just a way of saying the thing we're measuring. A variable is going to be any characteristic that can be different for different individuals. And we can have variables serve different roles in our statistical context. So we may have variables that are our response variable, the thing that we want to see what happens to. We might have variables that are our explanatory variables, right? So, and those are ones we think are going to have some effect on our response variable. So those are different ways we can talk about variables. And you might have heard of control, right? A control is sort of a special version of a variable. That's the sort of no treatment, right? But what we're measuring, whatever it is we're looking at, our individual, if we're looking at something where there's only a handful of different kinds, like outcomes on our dice, right? We're going to call it a discrete or categorical variable. And so we're going to have, like our dice, or sorry, dice is a bad example here. So like if we're flipping a coin, it has a heads or a tails, right? So we're going to count up how many heads we got. That's like putting them into a bin, right? So a category. Ordinal variables are like dice, right? They're numbered with whole numbers. We can't roll a die and get a 5.5, right? There's no seven, sorry, there's no seven at all. There's no 1.347365 right? So ordinal variables are numbered. They represent some kind of order, um, but they aren't necessarily going to act, they act more like discrete variables than they act like continuous or quantitative variables. Those are going to be variables we're measuring with your sort of things you think of as scientific instruments, and they're going to have some kind of decimal associated with the measurement. Theoretically, these variables can take on any value along the entire number. All right, so we could have that 1.736542 value if our instrument were precise enough. All right, so we're going to focus today on a continuous or quantitative variable and its distribution. So the distribution is what values our variable can take on or does take on, and how often it takes on those values. So think about our probability distribution graphs, and we were looking at the distributions in our so we will be looking at distributions of plant heights in our histogram activity, but we were using randomizing devices like dice or flipping coins when we made a sketch of the distribution of dice rolls, for example. That was showing what values, the possible outcomes, right, that the variable takes or can have, and then how often, right? So those proportions of rolls, that was how often, all right, that we come out with that particular value. And measurement is going to be an estimation of the value of our variable on an individual using an instrument. All right, so and scientific instruments have a lot of characteristics, right? We want to use ones that are standardized in some way, all right? So, and those kinds of units that we use in scientific measurements, centimeters, milliliters, degrees Celsius, those are the kinds of measurements that we're talking about. All right, so a quick note on significant figures and how they're going to work in this class. All right, so when we're doing statistical analyses, we're getting the data, like for purposes of this class, we're getting the data how it is. So somebody measured it using some kind of instrument, and the precision of that instrument determined how many digits after the decimal place. So when we are we've been given a data set and it has, say, 2 decimal places, all of the measurements should have the same decimal places, numbers after the decimal place. All right, so, and then we do some calculations. Say we calculate the mean or the standard deviation, and we actually get a number. Our calculator gives us a number that has 8 digits after the decimal place. For purposes of this class, we're going to round off so that our final number that we give as our answer to whatever problem we're working on is going to have the same number of digits after the decimal place as the data that we got. All right, because that is a representation of the precision of the instrument that took the measurements. And if we report a number with more decimal places, then we're misrepresenting the way the measurements were made in the first place. All right, so it'll take a little bit of a practice to remember to do that. All right, so when we look at our histograms, the thing that we are going to want to do eventually, ultimately, our goal is to look at this distribution. So what a histogram is going to be a distribution of how often or how frequently we have observed observations within a certain range along the x-axis. So this example graph, or sort of two graphs in one, is the distribution of blood pressure. And you may know that blood pressure comes in two kinds, systolic and diastolic, which represent whether the blood is leaving the heart or coming back towards the heart. So the blood pressures are higher, the numbers are larger for the systolic pressure, and they're lower for the diastolic. All right, but notice that we have a cluster of values on the left for the diastolic blood pressure, and we have a little bit more spread out group of measurements over here to the right-hand side that are the systolic measurements, right? The graph is representing, all right, perhaps thousands of patients that had both their systolic and diastolic blood pressures measured. And then, because the y-axis on this graph is percent, the people who made this graph took the proportion of individuals that had blood pressure in each of these little ranges across the bottom of the x-axis. Right? So they didn't put a tick on the x-axis here for every single individual. They did is they chopped the x-axis into these little bands or strips. Right, and I'm going to call those bins. Right, so we have a little section along the x-axis, and then they counted up how many of their observations fell into that little length along the x-axis here. So our histogram is basically going to be grouping together into bins how many observations we saw in that interval along the x-axis. And when we see how many pile up in each of these bins, it tells us how frequently each of those observations along the x-axis were observed in a sample, right? And so what we are typically going to see in a large sample of many biological variables is that we'll have this sort of characteristic bell shape, right? There's slightly different shapes on these two graphs. The one on the left is taller and skinnier. The one on the right is wider and lower. And they match pretty well with what the bars on the histogram are showing. So we'll call these bars, the heights of the bars, or sort of how far up they go on the graph. So when we've made a histogram, the first thing that we'll be checking for is, does this distribution we're seeing on our graph look normal? Now, this isn't saying, is it like the best individuals there are? Is it, or is it weird? This isn't a value judgment. And we're actually, this is shorthand. Are the data normally distributed? Right, the normal distribution is, there's a mathematical equation for it. We'll come back and talk about it a lot pretty soon, but it has this particular shape, right? And there are parameters that govern how pointy the top is and how wide the shoulders are relative to the tails, right? So when we look at the shape of a histogram that is fairly normally distributed, which these distributions are fairly normally distributed. All right, there's some differences, right? Some of the bars are higher, some are lower, but there aren't systematic patterns where these colored bars across their whole pattern are really don't match to this normal curve shape. All right, so our normal curve or our bell-shaped curve has some properties we're going to use to describe it. It's unimodal. That means it has one main bump, right? It has one main bump in its distribution. It doesn't have multiple peaks. Now, if we didn't know that these two sets of data were from different measurements, right? Systolic, we know systolic and diastolic blood pressures are different. So we wouldn't want to we know not to mix them together. But suppose we didn't know that they were different kinds of measurements, right? And we looked at this graph and we saw, well, there's a big peak over here, and then we have this sort of area that's lower on the graph, and then we have this other big peak over here. If we didn't know that these were two kinds of blood pressure measurements, we would see what we thought was a bimodal, or two modes, two peaks, right? We would see that distribution. Typically, a bimodal distribution is going to tell us that there are in fact two different things mixed together that should be separate from one another. So some other variable, in this case, what type of blood pressure is it, really is important in determining what the value of the blood pressure actually is. All right. So in this case, we have type of blood pressure is explanatory. It tells us why these two groups of values do have this difference between them. All right. So our normal distribution is unimodal. Right? Our normal distribution is also symmetrical. On each of these, if you look out to the right, and we're going to call this part of the distribution the tail, all right, and we look out to the left, and then we sort of go back to the center. Symmetrical is going to mean that the tail on each side, the left and the right, are about the same length. All right, so symmetry is going to mean when we go from the center of the graph to the left edge, and we go from the center of the graph to the right edge, that those are the same. All right, so our normal distribution is symmetrical. Mesocurtic is a word that's hard to define until we look at the things that are not normally shaped. So for now, just think about mesocurtic as being a shape that through the shoulders area here, the shape that's through this part of the graph, how wide it is at the shoulder. Is it the same as it should be if it's normally distributed? So if it's normally distributed, the shape of the shoulders, we'll call that mesocurtic, right? And then we're going to look for outliers. Outliers are going to be observations that are so far from the rest of the distribution that they look unusual. All right. Now, if we do have an observation that we think looks unusual, not part of the pattern, there's a couple of reasons that might be important. It might be a mistake we made putting data in, just a typo. It could be an individual that has, maybe it's the wrong kind. There's some other variable. We accidentally have a, measurement from somebody's dog in here instead of a human measurement of blood pressure. All right. So a normal distribution does not have outliers. We go out to the edges of these tails and the distribution stops, right? If we had a big gap and then we had some other observation way out here, that would be something we call an outlier. All right, so our normal distribution is unimodal, symmetrical, mesocurtic with no outliers. And what we're going to do when we look at all of our histograms in this class is we're going to check them to see if they match the normal distribution. And the reasons for that have to do with how our statistical tests are set up, right? So our data are going to need to be normally distributed in order for most of our statistical tests to apply properly to them. All right, so we're going to start practicing this. And the way to remember, because the most frequent thing students make mistakes on is that they don't write out an answer to all of these conditions. So if I say, is it normally distributed? You aren't going to say, yeah or no. You're going to say, well, professor, you're going to tell me how many modes it has. All right. And this acronym MONKS is going to help you remember. M is going to be for modes. The O is going to be for outliers. Does it have any outliers? The N is going to be this main question. Okay, our central question in doing these activities, describe the histogram, what's going on with it, is to answer the question, is it normally distributed? So the N is for normal. K is for the kurtosis. So part of this word, mesocurtic, the K there is for the kurtosis, and S is for symmetry. All right, so you want to ask the question, is it normal? Which is going to be the N, and then go through systematically and give me the M, the O, the K, and the S. All right, if it looks normal on those different characteristics, You still want to list them off. Well, professor, it's unimodal, symmetrical, mesocurtic, and it has no outliers. Okay. All right. So we're going to practice on a few histograms, just looking at these characteristics and what graphs look like if they don't have them. And then you'll be ready to describe histograms as part of your histogram activity. All right, so here's an example. And this one conveniently has this overlay of what the normal distribution should look like for this set of data. All right, so these are diameters of fairy shrimp eggs or cysts, and I had some students measuring these when I was working at the University of San Diego, because the fairy shrimp have a really interesting and complex life cycle, and the eggs can stay in a state of suspended animation for a really long time. And then, for some reason that we don't understand, they'll decide to hatch. So we were trying to look at the eggs and see if there were any differences in the eggs that might actually help us understand how the eggs knew if it was a good time to hatch or not. All right, so this is a sample of, so here capital N is the number of individuals measured. So I had a couple of undergraduate students busily at work on this project, and they measured 450 eggs. And then the mean of all the measurements was 33. This is in microns, and the standard deviation was 3.83. All right, so our mean of our distribution should be here in the middle, 33. So if we put a normal distribution on this graph, it should have its one mode right here in the middle. All right, however, looking at this graph, look, it has one big hump over here, a big dip down, and then a second hump over to the right-hand side. All right, so I would call this distribution bimodal. It has two peaks. And so it does not match to the normal distribution. If we look at its symmetry, it's fairly symmetrical. We don't see any outliers. There aren't any big gaps going on at the edges here. Because it's bimodal, it's a little difficult to tell if it has the same kurtosis as a normal distribution does. All right, but once we find one thing that causes it to be different from the normal distribution, then it's not normally distributed. All right, so this is a bimodal distribution. And so we have these eggs here, which are maybe a population of two different sizes of eggs, so maybe this is something that could be interesting to follow up in terms of, what are the differences between these eggs and how they behave. Unfortunately, I wasn't able to actually follow up on that research, so I can't tell you the answer. Okay, so again, here's a summary slide of what we're going to be supposed to do when we're examining our histogram. So our overall question is going to be, is it normal? We're going to look for deviations from the pattern, which are going to be outliers. We're going to look at its shape. Now, the answers you're going to have to these questions are going to be either unimodal, bimodal, like the one we just looked at. If it's got three or more modes, we'll call it polymodal. If it's not symmetric, it's going to be asymmetric, but we're going to describe it as either as being skewed. All right. So in assessing symmetry, it's not symmetric. You're going to need to specify if it's skewed, and if it's skewed to the left or to the right. If it's skewed left, it has a long tail to the left. If it's skewed right, it has the long tail to the right. Okay, make sure you're getting your left and your right straightened out, because that's an easy mistake to make. You'll literally see me holding my hands up in front of me and looking to make sure that I am labeling my left, my actual left. Okay, and the kurtosis, if there's no kurtosis, if it matches the normal curve, then we'll call it mesocurtic. All right. If it's too flat, right, if it's flatter than it should be, and it's sort of a humped shape, then we're going to call it platycurtic. Platy means flat. And then if it's more peaky, if it has like a steeper sides, shoulders than it should, we'll call it leptokurtic, right? So either it's mesokurtic, platykurtic, or leptokurtic. All right, so here's what those are going to look like, our non-normal distribution shapes. If it's skewed to the left, it has a tail kind of a less steep slope on its left-hand side. If it's skewed to the right, it has, typically we'll see it has a long tail to the right. If it's platykurtic, instead of having that nice normal shape, it'll seem kind of flattened and dumpy looking. And then leptokurtic, it's going to be skinnier in the shoulders than we expect. It's going to look too peaked or too steep in the middle. And you will get pretty good at recognizing these shapes, but it's going to feel weird to begin with. So we're just going to do a couple of practice graphs to make sure that you are kind of understanding how to use these vocabulary words. So this example is on shark length, right? So notice that what was measured was the length of the sharks. And when we look at these bins in the bottom, they're each 1.5 feet. Now it does bug me that this is in feet and not in meters. We have a gap here. That means there weren't any sharks measured in this 10.5 to 12 feet length. These just enormous sharks. We also have a gap over here. That means that there were no sharks measured between 21 and 22.5. All right, but notice that these, each of these green bars, the one on the far left and the one on the far right, only represent one individual. All right, so we look on the frequency to the left. All right, the highest bar here is going up to 11. So if we're looking at symmetry, first of all, let's look for mode, right? And we've got one main peak here in the middle, so it's unimodal. Symmetry, it's about the same distance to the end of the tail on each side, so we'll say it's symmetrical. It doesn't have any outliers. If we draw, okay, so try and draw in your mind or even with your hand a normal curve over your graph, right, like the ones that we've seen. And outliers are going to be ones that are really outside the normal expected path. So just because there's a space here, it just means there weren't any measured in that bin. All right, it doesn't necessarily mean that this observation is an outlier. Now, if we look at this graph, if you're thinking to yourself, this looks a little funny, it doesn't look really perfectly normally distributed. You're right, there's something weird going on here. Okay, so there's sort of two ways you could describe this graph. You could say it's bimodal if you really want to, if this was really an obvious shape to you, bimodal, that would actually be okay with me. Right, what sticks out more to me is that on this side, we have more of a shape on the right-hand side, and as we try and sort of curve down to the left, we find that it's the shoulder's too wide on this side. So you could also describe this graph as being slightly platykurtic, right? It's flatter or wider through the shoulders than we might be expecting it, right? So if it looks like it's not perfectly normal, there may be more than one reason for that you can explain, and you can give a lot of nuance here. You can say, well, professor, this looks like it's not perfectly normal. This is the problem here. I'm going to describe this as being bimodal. That's okay, right? You need to say something about the modes, something about all these different aspects. I'm not grading you on whether you would describe it exactly like I describe it. All right, we're practicing describing. All right, so do your thing. All right, so here is a different example of COVID incubation times. This is from 2021 paper, and we have across the x-axis, we have days, how many days it took for the virus to incubate. That is between the time a person was infected and the time they came down with symptoms. We do have one tall peak here at around 5 days, right? So now you could conceivably describe this as polymodal, right? It does have kind of these dips, all right? You could also describe it as unimodal. You could really sort of go either way on that, would be okay with me. Now, when we look at the symmetry, this is where we say, oh my gosh, that does not look symmetrical. All right, we should have a much longer tail to the left if it's going to match the length of the tail to the right. Okay, so we have an asymmetrical graph, and it has a longer tail of data to the right, and so that makes it right skewed. All right, so this distribution is right skewed. If we look at its shoulders, kind of this region here, does it look too tall and peaky through that region, or does it look maybe rounder and flatter through the region? I'm going with rounder and flatter. All right, so this sort of polymodal, possibly platykurtic shape distribution, those two things are often gonna go together. And we have a long tail to the right here. I wouldn't necessarily call this one out here at 26. I wouldn't necessarily call it an outlier because if we draw our distribution and our tail keeps going, it really isn't that unusual. All right, it would have to be out maybe 29 days, 20, 30 days before I would say that was a very strange observation compared to the rest shown on this graph. I'm checking in my mind. All right, overall, it's not normally distributed. Okay, so you want to always make sure that that is your either your starting or your final statement. All right, our last example here is to have you compare the histogram on the left-hand side and the histogram on the right-hand side. Right, so I'm going to give you a minute to look at them. Look at what they're graphs of. So the title's up here, cases of COVID-19 and deaths of COVID-19. All right, and these data are from 2020. And look at the distribution patterns on the graph on the left, the cases, and on the right, the deaths, and practice describing them. Pause the video. And then once you are ready, start it up again and you can check and see if you are correct. Okay, here we go again. So this graph on the left, cases, it's pretty evenly distributed when we look across from 20 to 29-year-olds. All right, there's possibly one peak, it could be bimodal, right? Overall, it's not normally distributed. It's really a weird looking distribution. And I would say definitely it's platykurtic. It's a little hard to tell if there's a skew because it isn't really normal enough that we have a lot to compare with. So sometimes if it diverges from normal enough, it's hard to evaluate some of the other characteristics. All right, so cases are distributed across adult age groups, all right, not uniformly, but in large numbers. Now, if we look at the deaths by age group, all right, this is really different. We see that a huge peak of the bar that's at the over 80. All right, so most of the deaths of COVID tend to be in the oldest age groups. So if we look at the distribution on this histogram that's on the right side and evaluate it against normal, we'd say, no, it's not normally distributed. It is unimodal, and it does have a reasonably mesokurtic shape if you kind of imagine that it was symmetrical, maybe a little leptokurtic. But it is strongly left skewed. All right, now I don't see any outliers on either of these graphs, but this left skew is often typical of sort of survival types of functions like this one. All right, so if we compare the histograms to one another, we see that depending on what variable we measure, we see a very different picture of how those COVID impacts are spread across the population. All right, so hopefully that gives you just a quick introduction to what are histograms, how are we going to describe them, and what are the kinds of research questions that they might allow us to answer. All right, so I will catch you next time.